Hello everyone, I welcome you back to another important video lecture series for your IGCSE Biology 0610 topical past paper solutions. And for today, we will look on toward all the topic questions from your chapter number 5, which is enzymes, and this is part 2. And in today's class, we would begin to be looking on toward the questions from the year 2016 and we will go till the end of 2025. But here the good news is that in the year 2016 and 2015, none of the question it appeared from the enzyme section. So we would begin from year 2017. So we would go till the end of 2025. The part one I left at question number 8, and now I begin from here from question number 9. And this appeared in your year 2017. This was May, June, and the variant was 4-3, and this was question number 2, D part for your past paper. And they say that we have got the researchers and they investigated the effect of the pH on the activity of the protease in quinoa seeds. Now, since I am looking on toward the effect of the pH, so what we are required to be saying is that this is a variable that we are required to be changing. So this would be acting as an independent variable. And the results that are shown in the graph that is given below. And if we were to see further, now you see on the x-axis we have the pH. This is the independent variable. The enzyme activity in terms of the arbitrary unit is shown on toward the y-axis. So this is something we are measuring. So this would be acting as a dependent variable. Now, if I were to be taking you toward the values, so you see between 2 and 2.5, each small box is representative of 0.1 increase in pH, so it is 2.1, 2.2, and so on. And if we were to look on toward the y-axis in between 0 and 0.1, we have again got small 5 boxes. So each of the small boxes is representing a value of 0.2. If I have to be describing this graph, so the simple idea is that at pH 2.5, the enzyme's activity is around 0.05 arbitrary unit. And then on all the given pH, the enzyme is working. But ultimately, when we are coming near to like 6.4 pH, so here the enzyme is no longer working because the graph has touched the x-axis. So this means that at this much high pH, the enzyme has got denatured. But the enzyme is more active in between pH 2.5 to pH 4. Why? Because the gradient of the graph is increasing. And you see that the pH 4, where the enzyme is exhibiting a maximum catalytic activity of 0.55, so the optimum pH is 4 with us. Beyond this, the explanation would be that here the graph is going downward. So when you are increasing the pH beyond the optimum pH, so the enzyme's active site they would start to denature and ultimately the substrate binding with the enzyme would be reduced and the enzymatic activity in terms of the arbitrary unit is decreasing. Now if we were to move toward the questions, what they say is for 2 marks, ask is that state two factor other than the pH that would affect the enzymatic activity. So we know enzymes activity is impacted by the temperature, by the pH, by the amount and the concentration of the substrate, but the pH they are changing. So other than pH, I am required to be keeping the temperature, amount or the concentration of either the substrate or the enzyme I have to be keeping it constant. Now here they are saying for the total of 3 marks that we are required to be describing the effect of increasing pH on the activity of the protease in guanine acid. As I have like clearly told you that how we were to be writing the description. So here I have just written the answer for this that the activity of the enzyme it increases from 2.5 to 4 and we have to quote him the values of the y-axis that this goes from 0.5 to 0.55. 0.05 not 0.55. You see here we have 0.05 and you see up till 4 we have reached to the maximum catalytic activity of 0.55. And this then further I have written that it decreases from pH 4 to 6.5 and I have given him the value that we are lowering the enzymatic activity from 0.5 and technically this is coming toward the 0. So we don't have to provide him with any explanation. And then we would say that beyond 6.5 pH, like beyond this pH, 6.4 or 6.5, you have the flexibility here. So in enzymatic activity, this remains 0. So this is how you were required to be writing the description for the total of 3 marks. Other than that, as an alternative valid point, if you forget any of the above or you don't see it in the graph or you're not able to elaborate it, so you would say that the enzyme is exhibiting the maximum catalytic activity of how much? 0.55. And this is achieved at an optimum pH of 4.0 that we are given here. Now here they are asking us for the total of 4 marks that you are required to explain the activity of the protease at pH 4 and pH 7. Now the explanation is coming, so we need to be writing the mechanism. So you see at pH 4 we have got the maximum catalytic activity. So here the explanation would be that maximum of the enzyme substrate complexes they are being formed, so that is why the optimum activity or the enzymatic activity is the highest. The frequency of collision at this point would be maximum between the enzyme and substrate. If an explanation is being asked, so what we would say is that beyond this 4.5, at this point of the graph, the enzyme's active site it would begin to be losing its shape. And when the enzyme's active site would be deformed, so the substrate would no longer bind into the enzyme's active site, and the enzyme would start to be denatured at a value of 6.4 and 6.5 pH. And now since they have indicated that you have to talk in 7, so we would say that at this 7 point, the enzyme molecule is completely denatured, the enzyme's active site is completely deformed, and no substrate is binding at this provided pH of 7.0. So if you were to simply see the answer, at pH 4, the explanation would be that the collision between the enzyme and the substrate is maximum, and that is why we see the optimum activity. At pH 4, the enzyme's active site is perfectly complementary to the substrate, and we would make maximum of the enzyme substrate complex. Here you have scored 2 marks. And then we would say at pH 7, the enzyme is completely denatured. The shape of the enzyme's active site is lost, and you see that when the enzyme's active site is lost, so no longer the substrate would bind into the enzyme's active site, and we would literally make no enzyme substrate complex. So if your answer would appear like this in terms of an explanation, so you would get total of 4 marks for this. Moving onward, this is a very simple question, and this came in the May June of 2018. The question number was 1A part, and you would say this is the variant was 41, and the course code is 0610. And here you see they have given us a diagram, and they say the reactions of the chemical digestion they are catalyzed by the enzymes. And you may have learned in your chapter of the animal nutrition or the human nutrition that whenever we are eating the complex food substances and when they go in our alimentary canal, so a lot of enzymes would be released and they would break down chemically the complex food substances into the simple ones. Now they have presented you with figure 1.1 and they say that we are showing you the stages of enzyme catalyzed reaction. Now if you would simply look on toward the diagram, this entire molecule that is given over here, so this is enzyme, and that location here that is presented with B letter. So this is enzyme's active site, and the A is required to be binding here. So A is the substrate with us, and you see that here we have got a complex of an enzyme and substrate. So C would be named as enzyme substrate complex. The enzyme is getting out of the chemical reaction as an intact molecule, and the substrate that we have here, so this has got converted into the product. Now, if we move to be seeing the question, so for the total of 4 marks, they have said that state the name of A to D, and as I have named it, that the A is substrate, B is the active site, C is the enzyme substrate complex, and D is the product with us. So this is how you were required to be writing the answer for this. Moving onward, we have got this question coming in October, November of 2018. This was question number 6B. The variant was 4.1. And they are saying that the digestive system of the young mammals, they are not fully developed. If there is a young mammal, he is like recently born. So the digestive system is not properly developed. And they are saying as that the enzymes such as amylase and the maltase and the protease, they are often added to the baby food. Now, why we are adding it to the baby food? Because their alimentary canal is not properly developed. So if we would add up the enzymes in their food, so they would accommodate in the digestive process. Now, they say is that complete table 6.1 by stating the substrate and the product for each enzyme reaction. So this was all empty. They have given you the enzyme here, then there comes the substrate, and at then we have the product. So if we have the amylase coming, the substrate of the amylase is starch, and it would be broken down into the maltose. If we have the maltase enzyme, it would act on a disaccharide, which is maltose, and it would be broken down into its respective two glucose molecules, but we are required to be writing just glucose for once. We have the protease enzyme, it would be breaking down the substrate protein into the product that we get out of the protein digestion, and they go by the name of amino acid. Now, further, they are saying for the total of 2 marks, they're asking is that suggests why the temperature of the baby food must be controlled when the enzymes are added. So we know that the temperature is a factor that would directly impact the enzyme's activity. So if the temperature of the baby food would be higher, then it would denature the enzymes in the baby food. And if the temperature of the baby's food is very low, so the frequency of the collision between the enzyme and the substrate would be lower. So for 2 marks, how you were required to be writing it down that the high temperature would denature the enzymes, because they did not say that high temperature or low temperature. They have given a general, you could say, elaboration that the temperature, why it needs to be controlled. So we have to write in both ways. So lower temperature, we would say that it would decrease the frequency of the collision between the enzyme and substrate. So I have written it in both in context of providing the high temperature to the baby food and low temperature. High temperature would denature it. Low temperature would decrease the frequency of collision between the enzyme and the substrate. For 1 mark, they say state one other condition that must also be controlled to optimize the enzymatic activity that we are adding them into their food. So there are a lot of factors. You see, except for temperature, we have pH. How much concentration of the enzyme there is, and how much concentration of the substrate there is. So for 1 mark, you have to write either one of these points. Moving onward to question number 12, and this appeared in the May-June of 2020. The question number in your paper was question number 2, variant was 41, and here is your paper code. And they say we have got the biological washing powders, and they contain the enzymes that break down the food stain. Like if I were to talk about the person and he was wearing some kind of a shirt, he was eating the food, and some of the stain got deposited on their cloth, and we know that this food stain would have the protein and it can have the fats in it, or it can have the proteins in them. So if we would wash this cloth with the help of the biological washing powder having enzymes in them, so ultimately this stain would be easily removed from here. They are asking us to be completing table 2.1, where we have to be naming the enzyme that would break down three substances that can be present in the food stains, and at then we are required to be stating the product or the products if they are made. Like if we just look on toward the table, we have the substance and there is this enzyme and the product coming. The starch, it would be acted upon by the amylase enzyme, and we would have the end product, which is maltose. The fat are always acted upon by the lipases enzymes, and we would have two products. One is fatty acid and the other one is glycerol. And that is why they clearly said either a single product or the products. And by the digestion of the fat, we are not getting a single product, but rather than we are getting two products out of it. If I were to talk with respect to the protein, it would be acted upon by the protease enzyme, and we would end up making amino acids out of it. If you were successful at writing the answers like this, you would have scored total of 3 marks here. Now moving onward, here are some of the students, and they try to compare that how effective the biological and non-biological washing powders are at removing the stain at the temperature between 10 and 60 degrees centigrade. Like they are fluctuating the temperature between 10 and 60 degrees centigrade. So this would be acting as our independent variable. Why independent variable? Because we would be changing it during the investigation. Now, what the students they would do that they would take some cloth, it would have stains on it, and then they would be adding it to the biological washing powder or non-biological washing powder they would be using, and into a beaker where they would have the cloth, and where they would have the biological washing powder, they would fluctuate the temperature to see that how effective the non-biological and the biological washing powders are at removing off the stains. Now, what they have said is that we have got the pieces of the stained clothes and they were washed using two different washing powders. The degree of the stain removal was measured by using a light meter to record the percentage of the light refracted from the cloth. Like I have got this piece of the cloth and this would have some stain. I would use some kind of biological or non-biological washing powder. Then we would have some kind of a light meter here and we would throw the light on it. And if all the 100% of the stains they would be removed, then ultimately the light meter it would have a value of 100%. That is an indication that the stain has been completely cleaned. They say any stain which is left on the cloth it would reduce the percentage of the light reflected. If biological or non-biological washing powder were not successful at removing the stain, then less of the light would be reflected and the value of the light reflected on the light meter would be less. Now here if we were to just look on toward the key, the dark line is showing non-biological washing powder and the dotted line is showing biological washing powder. And if we were to simply see that how we are required to be understanding the graph, so the temperature is on toward the x-axis, this is independent variable. The percentage of the light reflected after the washing, this is dependent variable because this is something we are measuring. If we see the distribution of the temperature, so here you, if you just simply see like in between 10 and 20 degrees centigrade, we have got total of 10 boxes. So each of the small box here, this would be representative of 1 degree centigrade. Likewise, if I were to come here in between 40 and 50 on toward the y-axis, the total small boxes are 5. So each of the small box, it would be representing a total of 2%. And you see that if you are given a key, so it is important that you just write it down or you just label the graphs in order to avoid the confusion. The dark one, this is representing what? The non-biological washing powder that we were using in order to be removing the stain out of the cloth. And the same, the dotted one that we have got. So let us just label it there and then that this is representative of the biological washing powder that we have with us. Now, if I were to simply describe the graph, you see that biological washing powder and non-biological washing powder, both of them, they are successful at removing the stains because we reach a value of 100% in case of the biological and non-biological. But the other idea is that at the temperature from 10 to 40 degrees centigrade, so the activity of the biological washing powder is great and the activity of the non-biological washing powder is less. The biological washing powder is successful at removing 100% of the stains or all of the stains because percentage activity is 100%. So this means biological washing powder, they are attaining their maximum staining removing ability at a lower temperature. But if we see for the non-biological washing powder, until or unless we don't raise it to a higher temperature of 60 degrees centigrade, so the non-biological washing powder would not be able to remove 100% of the stains. Another point is that if we just look on toward the temperature values from here to here, so we see a less of the difference between them. So this is an indicative point that when I'm trying to fluctuate the temperature from the total of 44 and I'm going till 60 degrees centigrade temperature, so we don't see much difference of the biological or non-biological washing powder in removing the stains. And at this point where both of the curves they are intersecting with one another, and that basically like is nearly around 43 degrees centigrade. So here the ability of non-biological and biological washing powder in removing the stain is same because we have got the same value of the light being reflected and that nearly like comes around 69.5. So if we were to see here, as I have explained above, so for the total of 4 marks, they say that compare the effectiveness of the two washing powders at removing stain, and we need to provide the information from figure 2.1 in our answer. So as I have explained above, we would say biological washing powder is effective at removing stain in a lower temperature range, and as I've shown you above, that the biological washing powder is very much effective at a lower temperature range from 10 to 30 degrees centigrade where it was able to remove the stain and the light reflected was absorption increased from 69 then coming till 100% that we have with us. Likewise, once we have quote him the value of the biological washing powder, then we have to compare it with the non-biological with the same range of 10 to 30. And where I have said that for the biological in between 10 to 30, we are reaching 100% of the light being reflected. But for the non-biological washing powder, if I were to zoom, so at 30 degrees centigrade here, the light reflected is 52%. So we have to be making comparisons. So that is why I am writing it down clearly that for 10 to 30, here the value for the biological is 100% light being reflective comparative to the non-biological washing powder that is reaching the value of 52% within the same temperature range. If we were to talk about the biological washing powder, so this removes the stains between 30 to 40 whereas a non-biological washing powder, it achieves it at a higher temperature of 60 degrees centigrade. But then, as we have seen above, that in between 50 and 60 degrees centigrade, we don't see any much of the difference. So that is why we would quote it him here that both the washing powder, no matter biological or non-biological, they have the similar activity at temperature between 50 to 60 degrees centigrade. So this is how, if you have written an answer like this, the total mark that you would have scored would be 4 for you. Now, if we were to move further, they say that the students suggested that the enzymes in the biological washing powders were denatured at a high temperature. And further, they want us to explain for the total of 2 marks that explain why the enzyme molecule do not function when they are denatured. So simply, like if I were to talk about the biological washing powder, and if we would increase their temperature to very max, so we know that the enzyme they would get denatured. And when the enzyme is getting denatured, then why it would no longer be able to be removing the stain because at such a high temperature, the shape of enzyme active site would be completely changed, and it would no longer remain complementary to the shape of the substrate molecule, and by that, no enzyme substrate complex would be formed. Moving onward, for the total of 2 marks coming here, they say forensic scientists they often try to find DNA on items of stained clothing. Like if we have got the stains on clothes, and the scientists if they are into some kind of an investigation, so they can find the DNA out of it as well. They say the DNA can be used to identify individual peoples, and further they ask us that suggests why the DNA can be used to identify the individual people. So if I were to talk about you or me, both of us we have the DNA, but in the DNA the sequence of the nitrogenous bases would be specific to you and to me. So if we simply try to be comparing this, so we would for sure have some differences among them. So for 2 mark we would say that the DNA in each of the individual person they have the unique sequence of the nitrogenous bases, and that too where in their genes, and it would be different among the individual people. That would make a difference between two of us or any other person to whom we are trying to comparing it with. Now, if I were to move to question number 13, this appeared in October, November of 2021. The variant was 4.2, and the question number was question number 1, and this was F part with us. This is specific to the enzymes. Now they are giving us some information about the funguses that they are decomposers and they would decompose the dead plants. And in decomposing the dead plants, these funguses, what they are doing is that they are excreting the enzymes and that would digest the large biological molecule that we have in the dead plants. Now, what the student they are doing is that again from the species of the funguses that are decomposing the dead leaves with the help of their extracellular enzymes. So they are making an extract out of it. And these extract out of the funguses, it would have some digestive enzymes. Now, what the student they are trying to investigate is that they want to be finding out that if amylase and the pectinase they were present in the fungal extract. So we would need to take some petri dishes. In one of the petri dish, we would need to be adding the starch. In the other petri dish, we are required to be adding the pectin because the pectinase enzyme would break down the pectin, and we would add the fungal extract into them. And as to be comparing it, we would take some of the, you could say, amylases and the pectinases from some standard solution to be making comparison that, okay, if the amylases or the pectinases, they were available or no. Now, what they have to be saying is that they made agar plates and they filled them with the help of the agar jelly and that either had the starch or the pectin in it. And they cut 4 holes of the same size in the agar jelly in each petri dish. Like if you just look here, we have got this petri dish. This has amylase in it. This is the other petri dish, and we have got the pectin substrate in it. Now, you see that these holes are made into these agar jelly plates, and into each of the section A, B, C, and D that we have. So we would adding some solutions into them. Again, what solution they are adding here is they are saying that for the hole that is punched at A, we are adding 1% amylase solution. Now, this amylase solution is not coming from the fungus. This is already there in our laboratory. Why we are including it along with the fungal extract to be making a comparison? Likewise, in the B hole that we punched here, so we are adding the pectinase enzyme solution into it. The C that we have here, so this is distilled water, And the D that we have here, so this is the fungal extract. Now, what they did next was that they kept all of these petri dishes for 4 days, and the temperature was kept as at 27 degrees centigrade. After this time, they poured a dye into each of the dish to stain the areas where the starch and the pectin remained. So they would add the dye here, they would add the dye here, and that is staining the petri dishes to be indicating us that where the starch digestion is left and here where the pectin remain undigested. They say figure 1.3 it shows drying of the stained agar in petri dishes. The clear zone indicates areas where no starch or the pectin was emitted. So if I were to explain it here, so we have the petri dish and this is having an agar jelly, but that agar jelly is having the starch. Now here we had 1% of the amylase. So the amylase is digesting the starch here. The B had the pectinase, you could say solution here, but since we don't have any of the pectin in the agar jelly, so ultimately the pectinase enzyme in B is not able to digest starch here because the shape of the enzyme active site would not be complementary. C is simply the water. And ultimately, the water you see is not able to break down the starch here, and neither this is able to break down the pectin here. If I talk about the D, so this is indicating that this D, where we added the fungal solution, this has the enzyme amylase in it, and that is why it was able to digest the starch. But if we just try to compare the zone cleared for A, that was 1% amylase solution, and the D that got cleared by the fungal extract, so here more of the area is cleared, and this is an indication that the fungal extract has more than 1% amylase concentration in it comparative to this. Now, we talk about this second petri dish, and in the second petri dish, the agar jelly it has a pectin in it. So this means that the water is not able to digest it. 1% amylase again would not be able to digest this pectin here. We have the amylase here, so the amylase cannot work on the pectin here. The B had 1% pectinase solution in it, so the pectinase is digesting the pectin here. The fungal extract again has got some A area clear, so this means fungal extract also has the pectinase enzyme, and that is why it was able to be digesting the pectin. But if we just try to compare it with the standard 1% pectinase solution here, so the area cleared by 1% pectinase is greater comparative to the pectinase which is released out of the fungal extract. So the idea is that the fungal extract has lower than 1% of the amylase, 1% of the pectinase concentration in it. Like the pectinase concentration of the fungal extract is lower, but that which we added as 1% pectinase, so this is higher. So this is all of the elaborations that I have given you as of now. Now, if I were to just simply show you the next diagram of that, so I have just simply labeled them up. And here they asked you for the total of, I guess they have a total of 4 marks. And they said that state one conclusion can be made about the enzymes in the fungal extract and give evidence from figure 1.3 to support your conclusion. Now, the general idea is that the fungal extract, it has both the amylase and this has the pectinase because in both of the agar jelly plates that we have with us, the starch is getting digested, the pectin is getting digested. So this is the first point that the fungal extract contains both the amylase and the pectinase enzyme. Okay, because the fungal extract is able to clear the zone of starch and the zone of pectin in both of the petri dishes. Then I would say that the fungal extract has higher concentration of amylase enzyme than 1% amylase. Like here we have the idea is that the zone declared is more comparative to this. So the fungal extract has more than 1% amylase concentration in it. And moving onward, if I were to talk about this agar dish, so I have said that the fungal solution has less concentration of the pectinase than 1% that we added here because the area cleared by 1% pectinase is greater and that cleared by the fungal extract is low. So this is how you would score total of 4 or 5 marks there were for this question. And if we were to move for the further 1 mark, they say that the investigation was repeated but at 5 degrees centigrade instead of 27 degrees centigrade. Predict the effect of the lower temperature on the result. So if I would reduce the temperature from 27 to 5 degrees centigrade, so the pattern you would say can be the same, but less zones would be clear. The idea is because at lower temperature, the collision between the enzyme and the substrate would be less, so less of the zone would be cleared. Moving onward, this question appeared to be in 2021 of your October-November and the variant was 4.2. And for the 2 marks, they are asking enzymes are catalysts. and they want you to be defining the term catalyst. So what we would say is that catalyst is a substance that would increase the rate of chemical reaction, and by doing so, it would itself not be chemically changed or modified. If we were to go on toward the B, they say that figure 6.1 shows diagram of three enzymes and eight different substrates. Okay, so we have this enzyme 1, this is enzyme 2, this is enzyme 3, and the rest of the letters that we are given over here. So they are representing total of 8 different substrate. They say state the letter of the substrate that will be broken down by the enzyme 1. So here I have the enzyme 1 with us. And the shape of the active site 1, we have to see the letter where this would be complementary. And if you see, this is kind of a circular active site. So the substrate T is complementary to it, so it would bind here. So we would write the letter T here. Now, moving onward for 2 marks, they are saying that we are required to be explaining in term of enzyme structure that why we have given the letter T here. So we would simply say that the active site of the enzyme 1, this has got the complementary shape to the substrate T we have with us. So that is why they both would make an enzyme substrate complex. So how you are right, we're supposed to be writing the answer, we would say that the shape of enzyme 1's active site, this is complementary to T. So ultimately it would bind with the enzyme's active site and we would make an enzyme substrate complex being formed out of it. Moving onward, here we have got total of 6 marks, and they say that table 6.1 list some enzymes and the reactions they catalyze. So again, this idea is we have got the enzyme here, the reaction that they are doing. So maltase is the breakdown of the maltose toward glucose, because maltase enzyme basically works on maltose, and maltose is a disaccharide having two glucoses in them, so it would be broken down to the glucose by the maltase enzyme. They say breakdown of the protein to amino acids, so we know this is protease. This is a general word for any of the enzyme that can break down the protein to amino acid. If you were to be writing specifically, so you could either say trypsin or you could also be saying pepsin. Moving onward, we have the lipase, and we know lipase, when they work on the fats, so they break them down into the fatty acids and glycerol. They say that breakdown of the lactose to the simple sugar. Lactose is a disaccharide. It has glucose and galactose attached with one another, and the lactose would be acted upon by the lactase enzyme. They ask us that insertion of the short length of the DNA into the plasmid. So the bacteria, they have got extra genetic material in them, that is the plasmid. And when we want to genetically modify a bacteria, so we basically add the gene of interest into this plasmid, and the enzyme would be the DNA ligase enzyme. And they say restriction enzyme. So the idea is that since I have isolated this plasmid out of the bacteria and I need to be adding the gene whose product I want to get out of the bacteria, I'm required to be attaching it here. So I need to cut this plasmid out. And this plasmid, if this has to be cut down into small fragment, and the plasmid overall is DNA by nature. So this would be the working of the restriction enzyme. Now, since we have to be writing down its role, so we would say that this restriction enzyme, this cuts down DNA molecules into the small fragments we say, and this way you would score 6 mark total of for this. For question number 15, appeared in May June of 2022, the variant was 4.1, and in your paper it was question number 1B and C part. And they say we have got some students and they were studying the activity of the yeast. And as far as this yeast is to be bothered about, this is a unicellular fungus. And for this yeast, the students, they have made a fact file, and that is being presented in figure 1.1. They say the organism is yeast. The scientific name is Saccharomyces cerevisiae. So this is the genus name. This is the species name. The structure of this is a single cell. And the main cell structures that we have in the yeast, they say this is cell wall, this is cell membrane, the vacuole, the nucleus, mitochondria, rough endoplasmic reticulum. So these are the major structures that we have got in this unicellular fungus with us. And they say main source of energy from where they would be aerobically respiring. So this, they are the sugars with us. Now, for 1 mark, they are asking, state the kingdom in which the yeast is classified. So the kingdom into which the yeast basically belongs from, like they have not asked the genus, but they are asking the kingdom. So the kingdom of the yeast, this would simply be the fungus with us. So we are writing this as fungus. And they say state the process that occurs in the mitochondria to provide energy in the yeast cells. So we know in the mitochondria, the aerobic respiration takes place. Now they say that the yeast cells they make the enzyme sucrase, and the sucrase would break down the sucrose into glucose and fructose. Like if we were to have this sucrase enzyme, and if the sucrose would bind into it, the sucrose basically has got two of the monosaccharides linked with one another. One of them is glucose, other than of one of them is fructose with us. So when they would bind into the sucrase enzyme, so we would end up making the glucose and the fructose out of it. They say enzymes are made up of proteins, and they're asking that how the shape of the sucrase molecule is related to the function. So what we know is that since sucrase is an enzyme molecule, so it has to break down the sucrose. So the shape of the sucrose active site, this has to be complementary to the shape of the substrate so that they both bind with each other to be making an enzyme substrate complex. And by that, the sucrose would be broken down into glucose and fructose, and by this you would score a total of 2 marks. They are saying that the student made an extract of sucrase from the yeast cells. Like I have got a lot of these yeast cells and I would crush them, and that would make me an extract, the resulting solution. And that extract has got an enzyme in it. And the name of the enzyme is the sucrase enzyme. Now, the sucrase that they have from the fungal extract, they are measuring its activity at the different pH values. So this pH that we would be fluctuating, it would be acting as our independent variable. Further, they say they determine the rate of reaction at each pH. So each of the pH which they have fluctuated, now they are calculating the rate of reaction. Now they say is they then calculated the rate of each reaction as a percentage of the fastest reaction, like that pH value where the fungal extract has given the maximum percentage, the rest of the activities at the pH, they would be calculated as a percentage out of that. If you just look on toward the graph, on the x-axis we have pH, this is independent variable. On the y-axis we have the percentage activity of the sucrase, so this is the dependent variable. If I were to go further, so you see in between 2 and 3 we have got a total of 5 boxes. So each of the small boxes is a representative of 0.2 increase in pH. And if I just simply split up the units given on toward the y-axis, so total of the 5 boxes are equivalent to 10 units. So each of the small unit here, this is representing 2. And now they are asking for the total of 6 marks that we are required to describe and explain the effects of pH on the activity of the sucrase enzyme, which they have shown you in figure 1.2. Now, as per the division of the mark is concerned, so 3 to 4 points should come for description and 2 to 3 points should go for explanation. Now, if I were to describe this graph for you, what I see is that 6 is the optimum pH, and here the enzymatic activity is 100%. From pH 2 to pH 6, the enzymatic activity is increasing and increasing from where 50% to 100%. Another point that I see is that in between say 5 and 6 pH, the increase in the percentage activity of the sucrose is steeper, and this increases from 82% and until comes till 100%. And if I were to just look on toward the graph from its range of 2 pH to 12 pH, so nowhere the graph is touching the x-axis. So this means that this enzyme is working at all the provided pHs which they have given you. But beyond 6, you see that the enzymatic activity is decreasing, and the explanation here would be that when the pH is increased beyond 6, so the enzyme's active site it would start to lose the shape. It would no longer be binding with the same frequency with the substrate as it was binding here. And if there comes an explanation point for this point here, right here at pH 6, where the catalytic activity is 100%, so we would say that at this point the frequency of the collision between the enzyme and the substrate is maximum, and that is why we are reaching 100% catalytic activity. So these are were the points for the description and explanation. So if I simply read it out for you, so I would say for description is that enzymes show activity at all the available pHs from 2 to 12. See, like from 2 to 12, the enzyme is showing activity at everywhere. And then I have said that the enzyme is showing the steepest increase in the percentage activity from pH 5 to 6 with an increase of 18%. So you see between 5 and 6, steeper increase from here 82% to 100%, and if we take the difference out of it, so that percentage increase is 18%. And then I have written as a description point that the maximum percentage activity of 100% is achieved at pH 6. And the minimum activity of the enzyme is seen here at pH 12, where the enzymatic activity is only 10%. So these are the description points. And if I go toward the explanation point, I have written in context of 6 that here the frequency of the collision between the substrate and the enzyme was maximum, and we have made maximum enzyme substrate complex. And beyond 6, the enzyme is slightly denatured, not like completely denatured, and the binding of the substrate with the enzyme is decreasing, and that is why the graph is going downward. Moving onward, for the question that came in May, June of 2023, the variant was fourth in your paper. This was question number 6 with us. They say figure 6.1 shows a photograph of a fruit which is attached to the branch of an orange tree. The genus is citrus and the species is sinensis. So if you just look on to here on toward its flower, so we have got total of, you could say, petals if we just count them up. So one, two, three, four, and five. So the if the petals say are four or five, or they are the multiple of them, so that is a dicotyledonous plant with us. So they are asking us for 1 mark. That's just one reason why the orange tree are classified as dicotyledonous plants. So 2 marks can 2 points can come here that the petals are five in the flower. And if I were to further zoom and show you the leaves, so here there are a lot of reticulations that we see here. So that again is for goes for the dicotyledonous plant. So since the marks are two, so you have to write any one from here. And they again say that the biologists they use enzyme to extract juice from the fruits such as oranges, and we are required to define the term enzyme for the 2 marks. So we would say enzymes are the biological catalysts. They are increasing the rate of reaction by lowering the activation energy. Moving onward for the C part, they have given you figure 6.2, and here they are showing the results of when we are leaving the pieces of the orange fruit in an enzyme solution for different lengths of time. Now, here they have given you the time in minutes, and here the total amount of the juice that is extracted out in terms of the cm cube. Now, here what you have to be keeping it in your mind is that if I have got the fruits and I want to extract juice out of them, so we are required to be using an enzyme here, and the name of the enzyme is a pectinase enzyme, because in the fruits we have the plant cells, and in between their cell wall we have got some gluing material, and that gluing material is pectin. So when the pectinase enzyme would digest this pectin here, so the juice extraction would become easier. Now they are asking for 1 mark that says the name of the enzyme that is used to extract the juice out of fruit. So the name of the enzyme is pectinase. Now here they ask again for 1 mark that we are required to use information in figure 6.2 to state the optimum length of time for efficient extraction of juice. And optimum length of the time mean where the graph is going upward. So if you see that when we are coming here at this point, so if I were to zoom more, so we have got total of 20 values in between small 10 boxes that we are given here. So here we have got 50, and if I just do the half of it, so the time here it would be 45 minutes with me. Until this 45 minute, the juice extraction out of the fruit is increasing, and beyond this it is becoming at a constant level. So the you could say the optimum length of time, this is actually 45 minutes. Beyond that, we don't see any increase in the juice that is being extracted out. So the answer would go as 45 minutes. Now they ask for the third point is that state the name of the vitamin which is found in higher concentration in citrus fruit such as oranges. So we know this is vitamin C with us. Now for the D part, this says that genes in some plants have been changed to increase the concentration of the vitamins that these plants produce. So they want you to name the process by which we change the gene of the plant. And we know it well that whenever the gene has to be modified or they have to be changed, so we employ the genetic engineering. Moving onward to 2022, this was October, November, and the variant was 4-1 in your exam. This was question 2 with you. They say is that the digestive enzymes, they catalyze the breakdown of the large insoluble molecules. And they are asking that why it is important that large insoluble molecules are broken down by the chemical digestion. So the protein and the sugar and the fats that we eat in our diet, they are the complex ones, and the complex food substances, they cannot enter into the cells. So the answer is very simple for 2 marks, and we would say that the large insoluble molecules, by chemical digestion, they become small and soluble. So they get absorbed into the blood, and finally they can reach toward the cells where they would be undergoing assimilation process. For the second statement here, they say state the name of the substance that is solvent for most molecules that have been digested by the enzyme. So after the enzymes they have digested the food molecules, the solvent that would be absorbing it, this would be the water with us. Now, if we just come on toward the B, they have given you the activity of the two proteases enzyme. One is A, the other one is B, and they measured their activity at different pH. So this pH, it would be acting as a variable, and we are required to be writing it as an independent variable. They say both enzymes they are found in the human alimentary canal. So what you need to be doing here is that you need to see on toward the x-axis. So from 1 until 6 we have the acidic pH, and then 7 is exactly the neutral pH, and then from 7 to 12 we have got the alkaline pH. Now if you were to recall your human alimentary can also, what we know from there is that the enzyme that works in the acidic pH, that is actually the enzyme A, which they have given in the key. So this is the pepsin enzyme that works in acidic condition and that are provided in the stomach. Now, if I were to talk about the enzyme B that we have here, and that is indicated with the dotted line, if you were to see, it is reaching its optimum activity at the pH of 10, and you have to be taking the readings of the B enzyme from here, and the A enzyme activity you have to take the values from here. So you see that this enzyme, since it's working optimally in the alkaline pH, so I can assume that this enzyme B is actually the trypsin that works in the alkaline pH in the small intestines. Moving onward, for the total of 6 marks, they want you to describe and explain the role of two proteases enzymes A and B in the alimentary canal. So as I've said, the enzyme A and B, they are the proteases enzymes. So their role would be to break down the protein that is in the alimentary canal to the amino acids. And from the graph, we have seen the enzyme A was exhibiting the optimum activity at the pH of 2, and this is 820 micron per milligram. Like you see, here, we have to talk in context of the role of the enzyme, and we have to provide the description and explanation. So at pH 2, this is how much the activity this is attaining at. And if I were to go further in terms of the explanation, in terms of the role, so we would say that why the enzyme, which is pepsin, is giving this much optimum catalytic activity. The reason is because this functions in the stomach, and in the stomach, the conditions are acidic due to the presence of the hydrochloric acid. Whereas for enzyme B, so we start, as we have seen from the graph above, so this is trypsin, and this is showing the maximum catalytic activity of 135 micron per milligram at pH 10. Now, if I just come here for the activity of the enzyme B, so you see is that in between 100 and 120, we have got total of 1, 2, 3, 4, 5, 6, 6.5 division. So if I were to divide 20 by 6.5, so every small unit here is representing 3.07 value in terms of U per milligram. So you see the boxes that I see from here is 1, 2, 3, 4, and 5. So when I would multiply 3.07 with 5, so this gives me 15.35. So I have to add it in 100. So likewise, for this, that is why we are writing that the enzyme B is trypsin, and it shows a maximum activity of 135 U per milligram at pH 10. And if we have to write an explanation for this, so one of the explanation is that this is getting denatured at 3. Why? Because this is preferring the alkaline condition, but 3 is acidic one, so it would denature the enzyme and it would no longer bind with the protein's molecule. And the other explanation for this would be that the trypsin works in the small intestine. The reason is over there the conditions are alkaline. Here you have to be bothering is that we were supposed to be describe and explain the role. So the explanation is coming in terms of their roles that we see for the enzyme A in case of the stomach and for the enzyme B in case of the small intestines that we have with us. Now moving onward, for 1 mark they say maltase is digestive enzyme that acts in the small intestine. They are asking you to tell him the exact location of the maltase in the small intestine. So in the small intestine we have the small finger-like projections, which are the villi. In the villi, we have these epithelial cells, and in the epithelial cells, we actually have this maltase enzyme with us. So For 1 mark, you have to simply say that these are the membrane of the intestinal epithelial cells where the maltase, lactase, and the sucrase enzymes they are available with us. Now let us come to question number 18. This appeared to be in October, November of 2022. The variant was 4.3 and in your exam this was question 2. They say digestive enzymes are excreted into the alimentary canal and we know that they are excreted. They say as well as enzyme, other substances that are important for the digestion are also excreted into the alimentary canal. So not necessarily the enzymes. Some other molecules are also being released into the alimentary canal and for 2 marks they are asking that share the name of these two other substances. So from the liver, the bile is coming. And in our stomach, we have the mucus. Then in the mouth, we have the saliva. And in the stomach, we have the hydrochloric acid. So I have written the name of four other substances other than the enzyme. So you're required to be quoting him any two from here. Now, again, they have given you the table for the total of 4 marks that this summarizes some facts about the enzymes that are secreted into the alimentary canal. So they have given you the name of enzyme, the organ where this would be secreted, the organ where the enzyme acts, and the product of the digestion that would be involving that enzyme. So if we have the salivary gland, the enzyme it would be releasing would be salivary amylase, and the organ that produces the salivary amylase, this is the mouth. And once the salivary amylase works on the starch, so either you could write maltose or you could say glucose. They gave you the name of the enzyme here, pepsin. So the pepsin is released, either you could say stomach or this is released by the gastric gland. And where this pepsin is being released into the organ, that organ is the stomach. And we know when the pepsin would be acting on the proteins, it would break it down into the amino acids. Then they gave you the pancreas here, and the product of the digestion, they are saying these are the fatty acid and glycerol. So this is simple that the name of the enzyme is lipase. And the organ where the lipase would be working, this is only the small intestines. They say this is small intestine and that is epithelial lining of the small intestine. So in the epithelial lining of the small intestine, the enzyme that we have with us is maltase. And when the maltase would work on toward the maltose, so we would have the end product made out of it and that would be glucose with us and by that you would have scored 4 marks here. Moving onward, again, they have said that the activity of the enzyme pepsin, it was measured at different temperatures. And they are showing you the result with the help of the graph. So if we were to see the graph, so on toward the x-axis, we have got the temperature coming. This is independent variable, and the enzymatic activity is shown in terms of U per milligram. So you see that as the temperature is being increased from 10 to 40 degrees centigrade, what I see is that the enzymatic activity is increasing. But this 40 temperature, or you could say 39 temperature or 38 temperature, either of the values, here the enzyme has reached the maximum optimum activity of 2500 U per milligram because this 40 or 39 or 38 is its optimum temperature. But beyond the optimum temperature, you see here the enzymatic activity has started to decrease. And at this 60 temperature, the enzyme has become completely denatured because enzymatic activity at 60 degrees centigrade has literally become zero. Now they ask us in terms of 6 marks that we are required to be describing and explaining the effect of the temperature on the activity of the pepsin. So the description is in terms of the quantitative number as we have done a lot of questions out of it. So the highest optimum activity is this 2500 U per milligram and this is attained where at 40. So this is the first description point. And then I have written that the enzyme's activity shows a steeper decrease in the percentage activity when the temperature is increased beyond the optimum temperature. Now, if we were to see here from 40 till 60, there is a steeper decrease in the enzymatic activity, and we would say that it reduces from 2500 and then ultimately coming toward the zero at this point. So this again is description. And now we would say that beyond 60 or beyond 61 or 62, you would say that the enzyme shows no activity at all because here the enzyme has completely become denatured. So the idea would be in terms of explanation that I have written in context of this point that as the temperature is increased, the kinetic energy of the enzyme and the substrate would be increasing and the collision between the enzyme and the substrate would lead toward more formation of the enzyme substrate complex with us. Another explanation you could give with respect to this point at 60, the enzyme has become completely denatured, the shape of the active site is lost, and the substrate are no longer binding into the enzyme's active site. So that is why the percentage activity becomes zero here at temperature of 60. So since it was description, 4 points should be in description, 2 should go for the explanation. So I have written 4 description and 2 explanation point. If we were to move further, They say that lactase is another digestive enzyme which is produced by the human babies. They are asking is that why the lactase is important for the nutrition of human young babies. So we know that the nutrition of the young baby is entirely dependent on the milk, and the milk has the lactose sugar in it. So once the babies would release the lactase enzyme, so that lactase enzyme, it would be acting on the lactose and it would break down the lactose into glucose and galactose that the babies could use for the respiration process, and ultimately the energy would be produced in them. For 1 mark, they ask is that most people they stop producing lactase when they become adults. If they drink milk, this may give them diarrhea, and they say that describe the symptoms of the diarrhea. The idea is very simple that people when they become adult and they stop producing lactase, so the lactose would not be broken down and they would develop the diarrhea. And the symptoms of the diarrhea is the loss of watery feces. If we move on toward this question that we have here, so this appeared in the year 2023 of October, November, and the variant was 4.2 and the question number was 4 in your paper. Now, here what they are trying to be telling you is that there are the scientists and they investigated the effect of the temperature on the rate of photosynthesis in one species of the plant. So we have got one species of the plant and we would be changing the temperature on that, and that change in the temperature, this is a variable that we are required to be changing in our investigation. So this would be acting as an independent variable for us. Now they are telling us that photosynthesis is involving some enzyme controlled reaction. So for sure, if the photosynthesis is happening in the plant, so the enzymes are involved. Now how the investigation was carried on, that they took the leaf from the plant and they took the discs out of them and then they exposed those discs to different temperatures. At the same time, all of the discs that they were involving, their surface area was kept the same so that it can act as a control. Now, moving onward, what they have to be telling us is that the volume of the oxygen that was produced by the leaf disk was measured. Now, we are measuring the volume of the oxygen gas to see that how come the temperature has impacted the rate of photosynthesis. And the more the volume of the oxygen gas would be produced, so that means that if we would divide that by the time, so we would have the rate of the photosynthesis. So this volume of the oxygen gas which is measured, so this is acting as a variable, and we would say this is a dependent variable. Now this is something that we are measuring. Now the results they are showing you in figure 4.1. If I just zoom this graph, so what you have to be seeing is that on the x-axis this is temperature, this is independent. The rate of photosynthesis in terms of arbitrary unit is shown on the y-axis. This is a dependent variable. On the x-axis, each small box is representative of one unit. Like here we have 21 degrees centigrade, 22, 23, 24, and so onward. And if we look on toward the y-axis, so again, each small unit that we are given here, so this is representing one arbitrary unit. Now, what they have to be asking is for the total of 1 mark that identify the optimum temperature for the photosynthesis, which is shown in figure 4.1. So you see that the optimum temperature is 40 because at this temperature we see the maximum rate of the photosynthesis that in this case is 37 arbitrary units. So 40 would be our optimum temperature. If we were to move further, so here they have given you total of 6 marks if you could see, and they want you to be explaining the results that are shown in that figure 4.1 above. Now we have to be explaining the results. We are not required to be describing it in terms of the numeric values. Now, if I were to just take you here to the graph in terms of the explanation, so you see that when the temperature is being increased from 25 to 40 degrees centigrade, so the rate of the photosynthesis is increasing and the graph is steeper. At this temperature from, you could say 35 to 40, the rate of photosynthesis is increasing but less steeper. So in this part of the graph, like from 25 to 35, what we would say that as the temperature would increase, so the kinetic energy of the enzyme and its respective substrate, they would increase. And as a result of that, the frequency of the collision, it would keep on increasing between enzyme and substrate, and we will be making more of the enzyme substrate complexes here. And until or unless at 40 degrees centigrade, where we have reached the maximum rate of the photosynthesis. So here the explanation would be that at this point, at this temperature of 40, the maximum enzyme substrate complexes have been formed, or all the enzymes that were available for this photosynthetic process, they have become saturated with the substrates on which they are acting. Now, if I were to present explanation from here to here, so you see that when the temperature is increased beyond 40 and coming till 50, so the enzyme is not completely getting denatured, but the rate of the photosynthesis is decreasing. So here the explanation is that as the temperature would increase, so the enzyme they would start to lose the shape of their active site. Okay, like the active site shape is not completely lost, but it's partially getting changed. So that would result in less substrate binding to the enzyme's active site, and that is the reason the rate of the photosynthesis is decreasing in this part of the graph. So here you see I've just explained it. If it was in term of description, then we would have given the examiner the reference with respect to these values and these values. So if I just read it out for you, what I have explained above, that when the temperature is increased from 25 to 40, the kinetic energy of the enzyme and the substrate molecule is increasing. And with an increase in their average kinetic energy, the frequency of the collision between the enzyme and the substrate, it would increase and we would make more enzyme substrate complexes out of it. And then as I mentioned above, that at 40 degrees centigrade, where we had the optimum temperature of the reaction, so the maximum number of the enzyme substrate complexes have been formed. And that is why at that 40 we had got the maximum point for the graph. But if we have increased the temperature beyond the optimum temperature of 45, so the shape of the enzyme active site it would begin to be losing and it would start to denature. And as a result, the enzyme's active site, when it would change, so it would no longer remain complementary to the shape of the substrate, and the collision between the enzyme and substrate would be decreasing. So this is how you were required to write explanation for the total of 6 marks. If I were to move further, so this happened to appear in the May-June of 23, the variant was 4-1, and in your exam, this was question number 1C. Now, what they have to tell us is about the action of the lipase, and we know the lipase would break down the lipids to fatty acid and glycerol. So, the activity of this lipase enzyme, this is impacted by the temperature. Now, they have given you this grid downward in figure 1.2, and on this grid, what the examiner is actually wanting that you are required to draw a line to show the expected effect of the temperature on the activity of the lipase. Like if we had the lipase and if we are increasing the temperature, then how come the temperature is going to impact the activity of this lipase? And then we have to add a label 9 to the same graph which they have given below. And over there, we need to indicate with a point that where the enzyme would be denatured. So you know that here we have temperature, here we have the enzymatic activity. So the graph should go like this in terms of the temperature, and it should become a little pointy here, and then you have to need to decrease it down. Because what I am assuming at my mind is that at this point we would have an optimum temperature of the enzymatic activity. So this is why with an increase in temperature, the graph is rising upward. But afterward, when the temperature would be beyond the optimum temperature, so the enzyme would begin to denature. And when the graph is completely touching the x-axis here, so this is the point where we are required to be indicating that here the enzyme it has got completely denatured. And by this, you would be able to score total of 2 marks. If I were to move further, so this actually appeared in the May-June of 2024. The variant was 4.2. And in your exam, this was question number 1C, and this was the fourth part of that. Now, what the scientists they are trying to investigate is the effect of sugar on the respiration that we have got in the yeast cells. Now, when we are saying the respiration in the yeast cells, so the idea is that the scientists they would have the yeast, they would mix them up with the sugar solution, and then they would be determining that how come the sugar is impacting the respiration in them. Now, in this investigation, they took two flasks. One of the flask it had 100 cm cube of the sugar solution, and the other flask that they included in the investigation, it did not have the sugar solution but 100 cm cube of the water. So what my idea would be that in the beaker or in the flask where we have the sugar solution, so if we would add the yeast into it and if we just block it with the help of the oil layer, so the yeast would be respiring anaerobically and we would be collecting the carbon dioxide gas here. But if I talk about the water, so if we add the yeast into the water, so the water cannot be acting as a substrate of the anaerobic respiration. And now further they say that both the flask of the sugar and the water, they have added the same amount of the yeast, and the temperature of that was kept constant at 25 degrees centigrade. And they have shown you the diagram of the apparatus which they have included. Like here we have the water bath and the temperature is maintained as 25. And here we have the like the yeast in sugar solution, or the yeast could be there in the water. And now they have placed a layer of oil on the top of it. Now that actually prevents the oxygen from getting exposure to the yeast, and then by that, the yeast here it would be respiring anaerobically, and by that, when the CO2 is being released, so ultimately the CO2 would start to collect here. If I were to go further, so they have given you the figure 1.1, and here they have indicated the results of this investigation. If you could clearly see, I have got two axes again. Here we have the time in minutes, and then here we have the volume of the carbon dioxide gas being produced in terms of cm cube. More the volume of the CO2 that would be released, that is proportional to more of the anaerobic respiration that would be taking place in the flask having the yeast in it. Now, for the flask where you had the yeast and the glucose in it, so the anaerobic respiration is higher because we have collected more of the volume of the CO2 until at this point where the graph is becoming constant. For the other beaker where we had the yeast and the water, so the CO2 is being released, like for sure the yeast would survive for some time in the water, and any of the glucose that were already present in the yeast cell before putting them into the water. So from there, the CO2 would be released. But after they would use it up, so the water cannot act as a substrate for their enzymes to be anaerobically respired. So that is why after 10 minutes, we don't see any increase in the carbon dioxide gas being produced. Now, if I were to take you to this question, and it has three parts, so this is the fourth part, I'm just including that is from the enzymes. They say that the scientists, they repeated the investigation, and they again took the yeast and they took the sugar solution, but now the temperature was increased to 95 degrees centigrade, and none of the carbon dioxide was produced now. If I just take you back, like initially the investigation was done with the help of the yeast and sugar solution, the temperature was 25, and we see a considerable amount of the carbon dioxide is being produced here. But at 95, no CO2 is produced because the idea is that yeast is living, and at such a high temperature of 95, all the enzymes that we have in the yeast, they would be denatured. The enzymes would lose the shape of its active site, and it would no longer remain complementary to the shape of the substrate. No enzyme substrate complexes would be formed to be making the carbon dioxide gas, and by that I have written a total of three statements in terms of explanation, and you would score 3 marks if your answer would have looked like this. To this question that we have with us, and this came in February, March of 2024. Variant was 4-2. And in your exam, this was the question number one with us, and they are telling you that the enzymes are the catalyst, and we are required to be telling our examiner the meaning of the term catalyst. And we know that the catalysts, they are the substances that increases the rate of the chemical reaction, and by doing so, they themselves would not be chemically changed or modified. Now, moving onward, they tell us that the enzymes are the protein, and we are required to be naming all of the chemical elements that would be available within the proteins. And we know proteins are one of the major fundamental biological molecule, and as per the elements are concerned, so it has the carbon and the hydrogen, oxygen and the nitrogen element. So by this you will score 2 mark. Here you have to be vigilant. They are asking the names, so you are required to be writing up the names, not their formulas. If I were to move further, they tell us is that lipase is an enzyme, and that is found in the human digestive system. What we know is that the lipase would break down the fats into the fatty acids and glycerol. Now they have given you figure 1.1, and they are showing you the effect of the pH on the percentage activity of the lipase. So the pH we would have changed, so it would act as a variable, and this variable it would be the independent variable. And since we are calculating the percentage activity of the lipase, so this for sure would be acting as a dependent variable because we would have measured it by changing the pH. So if I were to just simply zoom this graph more, so you see on the x-axis the pH is independent variable, And between 3 and 4, each of the small unit is a representative of zero point 0.2 change in pH. Likewise, if we just look on toward the percentage activity between 0 and 20, we have 5 boxes. So each of the small box is representing total of 4 units. Now, if I were to move further, so what they have to be asking is for the total of 6 marks that you are required to be describing and explaining the change in the percentage activity of the lipase, which is shown in figure 1.1. And by 6 marks, like there could be 4 descriptive point, 2 explanation point, or you could write 3 description and 3 explanation points. So let me just simply try to be describing the graph. Description simply means in terms of quantitative numbers. You have to give him the reference of the values on the x-axis and you have to quote him the y-axis values without giving any explanation of whether the enzyme substrate complexes are being formed or no. What I see is in terms of description is that at this pH of 3.8 that we have with us and 10.2 that we have with us, so the enzymatic activity is zero. This is description. And then I would tell him that when the pH is increased from 3.8 to pH 7, so the enzymatic activity has increased from 0%, ultimately coming toward 100%. Moving onward, the other description point is that pH 7 is the optimum pH, and here the enzyme has exhibited 100% catalytic activity. And then you would tell him another description point is to your choice that from pH 7 to pH 10.2, the enzymatic activity is decreasing from where? From 100% and then ultimately becoming zero at 10.2. So all of these they were the description points. Now, if we just write down the explanatory points, if I just tell you, so you would say that from pH 3.8 to pH 7, there happens to be an increase in the collision between the enzyme and substrate, and that is why the percentage activity of the enzyme is increasing. In terms of pH 7, you could write an explanation point that at this point, maximum enzyme substrate complexes are being formed. If you are writing the explanation from here to here, so you would say that as the pH is increased from 7 to 10.2, then the enzymes active site they would begin to denature, they would lose the shape, less substrate would bind into them, and that is why less enzyme substrate complexes would be formed. Finally, at 10.2, the enzymes active sites are completely deformed and none of the substrate binds here. So that is why the activity would be 0%. So all of what I have explained here, so let me just take you to my answer. As I have explained above, then enzymatic activity is zero at pH 3.8 and 10.2. And this is description point. Now I have written the explanation right in front of this that this is due to the reason that enzymes they get denatured at extreme of pH and this would cause the shape of the active site to change and it would no longer remain complementary to the shape of the substrate. Now, this was the descriptive point and then I have given the explanation for the point that I have written at first. And then as we have seen that the enzyme has shown 100% catalytic activity at its optimum pH of 7. So this is description point and the explanation for this would be that at this point of pH 7, maximum or most of the enzyme substrate complexes have formed. If you were to further write, you can write some other points as well. That we see a steeper increase in the enzymatic activity between pH 5 to 6, increasing from 24% to 80%. Like if you see from pH 5 to pH 6, there is a steeper increase in the enzymatic activity. So this is description, and from where this has increased, like from 24%. And if I just take you to 6 pH, so here it has increased from 24 to 80%. So this is description point. And the explanation would be that in between pH 5 to 6, the frequency of the collision between the enzyme and the substrate has increased. So this is the explanation for this descriptive point. Now moving onward to see first part for 2 mark, they gave you the name of the enzyme just and they left these boxes vacant. So I know is that the lipase react on the lipids and they convert them into their respective fatty acids and glycerol. For 3 marks, they are telling you is that the lipase would act in the duodenum of the small intestine. Now, what they are asking further is that you are required to be explaining that how the body provides a suitable pH for the lipase activity. Now, what we know is that the duodenum of the small intestine, this is receiving the bile from the liver, and that bile would make the content of the small intestines as alkaline because the enzymes in the small intestine they require the alkaline pH. Now, since the food has yet arrived from the stomach, and we know that the stomach would be sending the food with having some hydrochloric acid in it, so we would not neutralize the acid that is coming along with the food from the stomach, then the lipase enzyme cannot effectively work here. So for 3 mark, how your answer would be that the suitable pH for the lipase activity, it is provided by the bile, and this has come from the liver, or this actually initially gets released out of the liver, and then it would be stored in the gallbladder. So that is why I have written that this bile has come from the gallbladder into the small intestines. And once this arrives in the duodenum of the small intestines, so it would neutralize the food mixture that have come from the stomach having hydrochloric acid in it, and when the bile would be neutralizing the hydrochloric acid, the pH of the intestinal lumen, it would become alkaline and it would suit to the activity of the lipase enzyme. Now here let us come to question number 23, and this came in May June of 2024. The variant was 4.3, and it was question number 3, and the B part was from the enzymes. Now in figure 3.1 that you see here, they have shown you two, and they have shown you the enzyme molecule, and that enzyme molecule is here, and this is enzyme 1. And into the active site of this enzyme, when the molecule A would bind, so this molecule is making an enzyme substrate complex here. And when this would be broken down, so the enzyme one is remaining intact, but the substrate where that is molecule A, so it is being converted into molecule B and C. Now, later they say is that now we have added a molecule D that is shown here in figure 3.2. So you see that here we have molecule A that was previously being converted into product B and C. But now they are asking us that when we add this molecule D into the reaction mixture, then the formation of the rate of the product that is B and C, this decreases. So the answer is very simple because we know that this molecule D, this has a similar shape to the molecule A. So this D would compete with A to be binding here within the active site of an enzyme. So as a result of that, the binding of the molecule A would be reduced due to the presence of the molecule D, and less of the product B and C would form. So this is the same thing that they have asked here, that you have to use information that is given in 3.1 and 3.2 to suggest why the rate of production of the molecule B and C decreases after molecule D is added. So as I have explained above, that molecule D has shape similar to D, and this molecule D has a shape complementary to the enzyme's active site. So this D, it would compete with A to be binding with the enzyme's active site. And when less of the A would bind with the enzyme's active site, so for sure we will produce less of the molecule B and C. Now, what they are saying further is that the same enzyme 1 which they have shown you above, they changed their environmental conditions. And as a result of that, you see that the shape of the enzyme's active site is getting a lot of change in it. And now for 2 marks, they are asking that suggest two changes that could cause the effect on enzyme 1. Now, since the shape of the enzyme's active site is being greatly changed, so you would write increased temperature could be a cause, or you could say that we have increased or we have extremely decreased the optimum pH, like we have changed the pH from the optimum value, and that is why when the pH is increased beyond the optimum or decreased below the optimum, so that increase and decrease in the pH is changing the shape of the enzyme 1. So I hope you would be clear with the dimensions of the past paper that appeared from the enzymes chapter in the previous years, and if ever it would come in your exam in 2026 or 2027, you would be able to crack it like a pro. So best of luck for this chapter, and see you for the next topical video for the chapter that is the plant nutrition with us.